Hello guys, as we have discussed the two sections of the physiology of the GIT, and these are number one, we discussed about the GIT secretions, that what are the secretions that occur through the GIT tract, and we have also discussed about the hormones that are secreted through the GIT tract. So the third section of the physiology of the GIT that we are going to discuss today is digestion and absorption. We will discuss the digestions that occur inside the GIT tract and how the digested materials are then absorbed into the blood, and by which process and how they are absorbed. We will discuss this in this lecture. In food we have carbohydrates, we have lipids, we have proteins, we have vitamins, we have iron, and we will discuss the digestion and absorption of all these substances. So first of all, we are going to start from the carbohydrates. In diet, the carbohydrate is in the form of starch, which is a polysaccharide, a complex polysaccharide. So when carbohydrates is in the mouth, we have one enzyme for its digestions, that is salivary amylase. The activity of salivary amylase is activated by the chloride ion. So the digestions that is done by the salivary amylase, it is responsible almost 5% of the digestion of the carbohydrates. It means that out of 100% that we have taken the carbohydrate, 5% of its digestion occur inside the mouth by the activity of the salivary amylase. So when the food reaches to the stomach, in stomach we have HCl. So it inhibits salivary amylase activity. So the activity is masked by the acid of the stomach, that is of the salivary amylase. So there is no digestive enzyme for the carbohydrates in the stomach. So it means that there is no digestion occur of carbohydrates inside the stomach. 95% of carbohydrate digestions that is done by the pancreatic amylase. Pancreatic amylase, it acts only on 1,4 linkages, and the product we get is disaccharide or trisaccharide. We have discussed that food in carbohydrate occur in the form of starch. Starch is a polysaccharide. So that starch, that polysaccharide is digested by the pancreatic amylase at 1,4 linkages, because starch is polysaccharide. The polysaccharide structure of the starch, it is found by the 1,4 alpha linkages. So the pancreatic amylase, it acts on them and convert into disaccharide or trisaccharide, particularly in the small intestine at mucosal surface. So there we have some other enzymes which are called sucrase, lactase, and isomaltase, and these enzymes act on disaccharide or trisaccharides. So these enzyme acts at the level of 1,6 alpha linkages. The disaccharide or trisaccharide formed from the polysaccharide, they are converted into 1,6 alpha linkages. So these enzymes then act on them and convert them into monosaccharide like glucose, fructose, and these are the monosaccharides which are stored inside the blood or inside the cells, and these are absorbed from the cells into the bloodstream. So we have discussed that how polysaccharide is converted into a disaccharide or trisaccharide, and how the disaccharide or trisaccharide converted into a monosaccharide. Suppose it convert into a monosaccharide, we consider that as a glucose. So how intestinal glucose absorption occur, we will discuss now. That glucose is absorbed, how it is absorbed, we have already discussed its digestion. So here we have a cell, intestinal cell. It is come near to a blood vessel. At this side we have blood vessels, and at this side we have a lumen. So glucose from the lumen come inside the cell. It comes inside the cell. Along with it, one sodium also come inside. So we have a special transporter for them that is called sodium glucose co-transporter. This bring glucose and sodium inside the cell. This co-transporter or this channel is called SGLT1. This is a channel protein in intestine for the glucose and sodium absorption. So all these co-transporter SGLT1, all these co-transporter are considered as the secondary active transporter. Suppose whenever you are giving a solution which is rich in glucose from outside, so along with the glucose, sodium will also absorb because both have a same co-transporter. Suppose we give ORS solution, oral rehydration solutions for diarrhea. So exclusively act at SGLT1 protein. These are absorbed exclusively at SGLT1 protein. So glucose enter to the cell. Now it moves to the blood vessel. So from the blood vessels we have an important transporter and that is called GLUT2. This is a transporter that brings the glucose from the cells to the blood vessels. So GLUT2 is a carrier protein, so it has done its job by a facilitated diffusion. This glucose is diffused into the blood vessels. The diffusion is facilitated diffusion. It requires energy to bring this glucose from inside the cell to the blood vessels. So along with the glucose we also discuss about the fructose. So fructose is absorbed by the GLUT5. GLUT5 is responsible for the absorption of the fructose. Same is the case, fructose is absorbed from the lumen into the cells, and then this fructose is transported from the cell into the blood vessels. So it is also the absorption of the fructose, it is also done by a facilitated diffusion. Now after carbohydrates absorption and digestion, we are going to start about the lipids, how the digestion of the lipids take place and how the products that are formed by the digestion of the lipids, they are absorbed into the blood vessels. So in diet they are found as a triglycerides. Lipids are found as a triglycerides. So at the mouth level we have one enzyme that is a lingual lipase. It do the digestion of the lipids. In stomach we have enzymes that is called gastric lipase. It does the digestion of the lipids inside the stomach. So in pancreas we have the most important enzymes called pancreatic lipase, and this pancreatic lipase, it is supported by a colipase and cholesterol esters. They do the digestion of cholesterol. So as a result of the act of the pancreatic lipase, colipase and cholesterol esterase, so the lipids are converted into monosaccharides like free fatty acids, cholesterol and fat soluble vitamins that are vitamin A, vitamin D, vitamin E and vitamin K. Now all these three, that is free fatty acids, cholesterol and vitamins, they are incorporated into a structures which is called micelle, and this micelle is considered as a transport vehicle. Now suppose this is a micelle which is consist of free fatty acids, cholesterol and vitamins. Whenever this micelle come close to an intestinal cell, so inside the cell we have low free fatty acids and outside we have greater fatty acids. So these free fatty acids from the outside, they will move into the inside of the intestinal cell through a passive transport. Remember, absorption of free fatty acid, it is always through the passive diffusion. So whenever these free fatty acids or this micelle is absorbed into the cell, so these free fatty acids in the intestinal cell, they are re-esterified into a triglyceride, as we have discussed before. They are re-esterified into triglyceride. So this triglyceride that is re-esterified inside this cell. This is incorporated into a structure which is called chylomicron, and this chylomicron is get transported. Now we will discuss about the digestion of the lipids. So for digestion of the lipid, we need a bile. Bile is needed for the digestion of the lipids. This bile, it comes from the liver, it is synthesized inside the liver, and then it come out, and it is necessary for a digestion of the lipids. When this bile salt, it release from the liver and it come close into the intestine, and it enter into the intestine exclusively at the level of the terminal ileum. This bile get absorbed and transport back to the liver. This process, the coming of the bile into the intestine and it again absorbed and go back to the liver, this process is called enterohepatic circulation. Bile synthesis, it is a totally energy dependent. Why bile is reabsorbed from the intestine and go back to the liver? So for the question that we have raised, and the answer for this question is because it is totally energy dependent. So in order to reduce the energy demand, in order to reduce the usage of the energy, the recycling is needed. So recycling of the bile, it occur almost 6 to 8 times per day. Now we are going to know about a short topic, which is short chain fatty acids. So short chain fatty acids, they are mainly found at the level of the colon by the action of bacteria, that is colon bacteria. Short chain fatty acids, they are 3 in number. Number first is propionate, number 2 is butyrate, number 3 is acetate. What are the functions of the short chain fatty acids? Number first function is they have a trophic action. They increase mucosal growth at the level of colon. Number second, absorption of short chain fatty acid is always in exchange of hydrogen ion. So they do best role in acid base balance at colon level. Number third function is they also prevent inflammation at colon. So this was all about the digestion and absorption of carbohydrate and lipids. So if you are new on our channel, please subscribe our channel.